So why do you think conservation is important?
Maybe it's because it's where the animals live and it makes them sad,
Or is it because of the oxygen in the air?
or maybe just because it looks cool. What do you guys think? Anybody have an idea? Nobody? All right, good thing we're going to tell you.
So my name is Ray Risen,
I'm Trey Hillier.
and we're here to talk about why conservation is essential to the future of medicine. So to begin, the benefits that the Amazon provides, the main purpose of our change project is we want to establish and make it so that the Amazon is not getting destroyed and we can use it for future research for medicine. Just some benefits that the Amazon already provides to us is that it is a carbon sink. So basically it absorbs all the carbon in the air. The plants do take it in through the roots and then turn it back into oxygen and re-release it back into the air for us to use. And then also, there's a lot of plants there that are already being used by indigenous people to cure illnesses such as inflammation, infection, deficiencies, cancer, and hyperglycemia. So then, the destruction of the Amazon has been present throughout the whole time we've been on the earth. And the main ways it's destroyed is from natural disasters, cattle, which is just dealing with them building pastures, and then deforestation via logging for them trying to get resources to make paper and whatever else they need wood for. And then building up infrastructure. The building of a dam that they're trying to build in the Amazon, it required 370,000 acres, which is a lot of land for them to destroy because they have to clear the way to the actual river to make ways for the equipment to get in there. And then this graph just kind of shows where the majority of the destruction is coming from, where it's coming from pastures, logging, fire, because in recent ways, the main natural disaster is fire, and it's been in the news more last year than it has anywhere, but it happens every year. It's just last year was the biggest fire that happened in the Amazon. So then basically the destruction of the Amazon leads to habitat fragmentation, which is basically when the habitat is broken up into different quadrants or zones, and it doesn't allow the animals to get the necessary resources or food that they need to survive. So then they overconsume a lot of the plants and animals and kill them off, and then there's not as much biodiversity, which leads to decreased biodiversity all throughout the Amazon. And then it also just leads to a decrease in resources for us to use and also for the animals.
So I'm going to talk about how the environment is actually involved in medicine. So this picture here, the praying mantis, and this wasp over here is actually from our own Dr. Miller, where he talked about how both actually naturally make a chemical that has antibiotic properties, which we're trying to research and isolate now. That's actually kind of how the first antibiotic came about, was through mold in bread. So from Alexander Fleming's discovery and development of penicillin, they state that the discovery of penicillin was from Alexander Fleming in 1928, accidentally actually. So what he noticed as he was sorting through some old petri dishes was that there was some mold in them, and it had a zone of inhibition. So a zone of inhibition, which is shown here, is basically just a zone around any type of chemical, in this case mold, which inhibits growth. So he found that he was able to isolate this chemical and replicate it, shown here as penicillin, and it was able to kill bacteria such as Streptococcus, Staphylococcus, and Diphtheria bacillus. So if we have stuff like penicillin that kills bacteria, why do we need more? Well, bacteria actually develop resistance through evolution. Through this graph here from EMED, you can see that penicillin specifically actually developed resistance within roughly 5 years, as with most of the ones, especially more recent, with exceptions of erythromycin and vancomycin being that they are sort of last resort antibiotics. Funny thing is this resistance was actually predicted by Alexander Fleming himself. So he won the Nobel Prize for the development of penicillin, and during his speech he stated this: "The thoughtless person playing with penicillin treatment is morally responsible for the death of the man who succumbs to infection with a penicillin-resistant organism." So how does this resistance occur? Mostly through overuse. They're often overprescribed, whether it be in agriculture or maybe just a needy patient demands some type of medication, or through misdiagnosis. Obviously, if you have the cold, which isn't a bacterial infection, antibiotics aren't going to do anything for you. improper use, so not taking the entire regimen of a prescription. And that's kind of what I show here from top 10s. So say you have a bacterial infection shown here, and you have a strand within that that's a little bit more resistant. So as you take your antibiotics, you start to feel better, so you're like, oh, I'm not gonna take the rest of these, I don't think I need to. Well, those more resistant strands survive because they needed longer to, get killed, and they eventually replicate until that's the only type of strain you have in your infection next time, which is gonna be more resistant, and your antibiotics are not gonna be as effective. The way that bacteria actually replicate and exchange this resistance is through horizontal gene transfer, which is explained by Prescott in the textbook Microbiology from 2008. The mechanisms of horizontal gene transfer include conjugation, transformation, and transduction. Primarily conjugation, which is the exchange of plasmids, which is just a chromosome, a bunch of chromosomes, almost like a small DNA sequence. So I have a short video to kind of explain that, because it's not that complicated, but videos.
Keep the phone you love. Well, the real phone you love, with a little help from Google Fi, a phone plan by Google.
People commonly understand how major plants and animals exchange genetic information, but the ways in which microbes like bacteria do something similar are often overlooked. Bacteria rely on several different mechanisms for the exchange of genetic materials. In one case, a bacterial cell may transfer genetic information directly into another bacterial cell through a process called conjugation. In conjugation, one bacterium grows a duct called a pilus, which attaches to the other bacterium. A genetic element known as a plasmid is then passed through the pilus from the donor cell to the recipient. In another case, viruses play a role in genetic.
So that's conjugation. Transduction, it's a way of genetic variation, but it's not as effective in vivo, so I'm not going to go over that really. So how can we prevent resistance? We can, a theory from McKenna from 2015 in her TED Talk explains that we can require agriculture to give up antibiotic use, being that the 80% of the antibiotics in use actually are for agriculture. Now, the antibiotics used don't actually treat anything. They're actually just used to prevent infection because of the harsh living conditions. At the very least, you can just not buy products that have antibiotics in them. We can double check all prescriptions to make sure that there's no misdiagnosis, or make sure no one's just giving out prescriptions just to get rid of like a mom that's being, annoying. We can monitor where resistance is occurring. That way you can kind of isolate that strand and get research started on it immediately. And we can also develop new research methods. So like we've been doing, we can steal from nature by researching how organisms develop their own type of antimicrobial agents. We can use antimicrobial peptides, that's shown here in the research done by Louis et al. From 2016, as they increase this peptide along with azithromycin, the concentration or the amount of bacteria actually decreased, showing that it's effective. We can use nanoparticles. So nanoparticles are just small particles of any type of element. So in this case, they used silver from Sondi's research. It actually inhibited the synthesis of a membrane protein, allowing for nutrients to just be unregulated on the flow in and out, preventing compartmentalization. And then CRISPR is a way to genetically modify organisms by exchanging or removing genes. The research done by Joseph et al. In 2015 removed resistance genes from a bacteria, showing that all of them were resistant to streptomycin and gentamicin. And lastly, bacteriophages. I want to talk about them specifically in their own slide because they're a little bit of my favorite and they're of specific interest to our cause. Now, bacteriophages only infect bacteria. They have no toxicity issues in eukaryotes because of this. So you can infect anything from a fungi to a dog to a human because of this. So there is a catch. They are very host cell specific because of endonucleases. Endonucleases, as stated from Voet in the Fundamentals of Biochemistry from 2016, these are basically enzymes that degrade foreign DNA. They identify that this is not a DNA sequence from our cell, so I'm going to degrade this. I don't want to be infected. So what we need is to find phages that have similar genetic sequence to the ones of bacteria we're trying to infect. To do this, we're going to need to have areas of high biodiversity in order to do this research and this bioprospecting. This can be done in rainforests and in oceans, but rainforests specifically because of their extreme biodiversity.
So then, the main thing we want to get from nature is be able to create new drugs that are derived directly from nature. In the United States, the top 150 prescription drugs, at least 118 of them are based off of natural sources that are found out in the wild. 74% come from plants, 18% from fungi, 5% from bacteria, and then 3% from vertebrate species such as snakes and frogs. So basically with that, we want to be able to do what we talked about earlier with the praying mantis and the wasp, and that, about how they have those agents in their body able to fight against bacteria. We want to be able to take those and be able to turn those into drugs that we could use for people. Less than 1% of the tropical trees and plants have been tested by scientists. So that's not a whole, the Amazon rainforest is massive, and for only 1% of it to be tested, we don't know what else is out there, and what other plants and animals are out there that we could benefit from, and be able to use their agents to fight off diseases. So the Amazon's role in it would basically be to provide the natural resources needed to create more medicine. The U.S. National Cancer Institute identified 3,000 plants that are active against cancer cells, and 70% of them come from the rainforest. So basically with that, it provides more resources, and we can be able to find a more and better cure for cancer. And also, a lot of the plants haven't been studied. So we would want to be able to research the plants, determine if they can be used for more medicine, and also be able to create more medicine to fight off illnesses. So our plan basically for this is we want to be able to prohibit the destruction of the Amazon to further research. We want to set up research zones. Since the Amazon is so big, we want to be able to break down the Amazon into different quadrants. So that way we can set up research stations and they can take on that one quadrant at a time and be able to get more back from the Amazon. We want to be able to bring plants and animals back from the Amazon rainforest, be able to study them in the United States. But in order to do this, we'd have to clear national security and get clearance from them. But while we're doing this, we'd like to build a research facility, and with the research facility, we'd like to be able to mimic the conditions of the Amazon and be able to regrow those plants here so we can mass produce those plants that we find effective. And then with that, we would be able to create different medicines that will fight off illnesses. And then we want to eventually be able to sell the new medicines to be able to fund our future research and fund more research with Amazon. With this, it would open up a lot more jobs for research for the people researching the plants down in the Amazon and also doing the research back in the United States in the facility. And then there'd be the gathering with them, trying to bring that strain of plant or animal back and making sure they don't get hurt while they're being transported, they don't get killed, or like the plants don't have something that goes wrong with them. And then production, there'd be people that would have to watch over the plants, make sure they're growing right, produce the medicine, and also sell it. And then with that, we need transport, they'll transport the plants and animals and organisms back here and also transport the new medicine out to different facilities. So in conclusion, with our change project, we want to be able to save the rainforest and make sure that it's, people stop destroying it, and we want to be able to develop more medicine for people to use. Since we have been developing more resistance to drugs, we want to have more medicine out there that can cure diseases so people aren't getting as many illnesses. And then create a facility to conduct the research, and then also just want to be able to create more medicine.